Good morning, everybody. I'm Nathalie Heuze, and I would like first to thank the organizer for giving me the opportunity to present the result of the Pseudomonas project. The Pseudomonas project is the result of a public-private partnership and aimed to develop an inhaled phage treatment for acute Pseudomonas respiratory infection. As you may know, respiratory infections represent the fourth cause of death worldwide, and almost 16% of pneumonia are due to Pseudomonas aeruginosa. Infection to Pseudomonas aeruginosa arise often in patients under mechanical ventilations, who are the target population for the Pseudomonas project. Pseudomonas infection also arise in patients with chronic disease such as CF or COPD. Treatment of Pseudomonas aeruginosa has become challenging over the past decade due to both natural and acquired resistance of this bacterial species to antibiotics. Presently, it is considered that approximately 20% of Pseudomonas aeruginosa strains are multidrug resistant, and the bug has recently been classified as a priority 1 pathogen by the organization. Considering the mortality attributable to antibiotic resistance and the cost for the society to the management due to the management of people with antibiotic-resistant respiratory tract infections, alternatives to antibiotics are urgently needed. Bacteriophages are natural viruses of bacteria. They infect only bacteria and are highly specific of the bacteria species. Once their genome is injected into the bacterial host, phage can either enter into a lytic cycle associated with virus replication and bacteria killing upon phage release, or enter a lysogenic cycle integrating their genome in the bacterial one. Lytic phages are those intending for therapeutic purposes. The majority of therapeutic phages belong to the codoviral order, which consists of three different families: Myoviridae with a long contractile tail, Podoviridae with a short tail, and Cyphoviridae with a long non-contractile tail. Bacteriophages are interesting because they can self-replicate as long as the bacterial host is present into the environment. Phage therapy has been successively described to treat respiratory infections, both in preclinical study, but also human case reports also describe success of phage therapy in different types of patients. The global objective of the Pseudomonas project was to develop a drug and device to deliver a phage cocktail in the airways to treat bacterial infections due to Pseudomonas aeruginosa during ventilator-associated pneumonia. The first step aimed at developing a phage cocktail to cover most Pseudomonas aeruginosa strains. So for that, we used an international reference panel and natural phages purified from various sources. We tested the sensitivity of bacterial strain to phages using two different methods, the spot method and the liquid method, and defined the best cocktail using a combinatory approach. Overall, we selected 5 phages that have 95% efficacy against the Pseudomonas aeruginosa species. 3 of these phages were Myoviridae, 2 of them were Podoviridae. During the selection process, we also considered the production level and the stability of each phages over time to select the best phages. The production and the purification were optimized to reduce as much as possible endotoxin. Next, we determined the sensitivity of the Pseudomonas aeruginosa strain Paclux which was intended for all animal studies, to the different phages. We found that Paclux was sensitive only to 3 of the phage out of the five that were selected, as you can see in this graph. A mixture of these 3 phage was able to rescue mice from death by Paclux after local delivery at a multiplicity of infection of MOI 100 or MOI 10, meaning for MOI 10 that there are tenfold more phages than bacteria. Overall, during this first part of the project, we were able to characterize a phage mixture that was effective on most Pseudomonas aeruginosa strain and against the Paclux strain intended for animal experiment. We also showed that the pulmonary route was more efficient than the IV injection of the phage cocktail to treat respiratory infections due to Pseudomonas aeruginosa. I did not present you this result today. We showed that a unique administration of the phage mixture at MOI of 10 was sufficient to hinder lung infection progression in a mouse model, and we also checked that phage therapy was equivalent to treatment with a reference antibiotic. To help bridge the gap between the mouse result and clinical trials, we next developed a drug-on device to deliver the phage cocktail by inhalation in human and to test it in an animal model of VAP that was closer to human, a porcine model that I will present you later. So now I'm going to discuss these two parts of the project. And first, I'm going to talk about phage inhalation and the development of the drug-on device. As you know, drug aerosols can be generated either as a liquid or solid preparation. Dry formulations are often considered for phage inhalation. But in our case, because the final application was to deliver the phage cocktail to patients under mechanical ventilation, we consider nebulization to deliver the phage cocktail. We assessed the impact of different types of nebulizer on the viability of phages. We tested a pneumatic nebulizer, an ultrasonic nebulizer, and 3 different mesh nebulizers, in particular a prototype static mesh nebulizer from DTF that was developed specifically for this project. As you can see in this graph, the phages had different resistance to aerosolization depending on their morphotype. The Podoviridae with the short tail resisted better than the Myoviridae to the different type of nebulizer. The mesh nebulizer was a device with the best output and survival rates. Overall, the residual intact phage in the cocktail after nebulization was 23% for Myoviridae and 47% for Podoviridae. The large discrepancy of viability between Myoviridae and Podoviridae indicates that the nebulization process can cause different type of damage on phage depending on their morphotype. This is what we analyze next by electron microscopy, looking at phages before and after nebulization. As you can see here, the structure of Podoviridae is intact after nebulization, in comparison to Myoviridae, where that have a lot of morphological damage after nebulization. Considering that only phages with filled capsid and attached to their tail are viable and infectious, the sensitivity of Myoviridae phages during mesh nebulization may be attributable to their fragmentation during mesh nebulization. As a brief summary of our result in this part, we found that the 2 main parameters impacting phage viability during nebulization are the morphotype of the phages, with myoviridae being more sensitive than podoviridae to nebulization, and the category of a nebulizer, with a mesh nebulizer most relevant for phage delivery based on their output, but also on the viability of phage during nebulization. The last part of this talk addresses the animal model of VAP and the efficacy and the fate of the phage cocktail in this model. We developed a pig model of acute VAP, due to Pseudomonas aeruginosa in this project to address the fate and efficacy of the phage cocktail delivered by mesh nebulization. The main advantages of large white pigs are that they disclose respiratory parameters and lung anatomy that are similar to human. Second, they are often used for aerosol delivery during mechanical ventilation. And recently, the team of Libasis in Spain has described and developed a pig model of VAP due to Pseudomonas aeruginosa. The pig model of acute VAP due to Pseudomonas aeruginosa is presented here. Prior to the experiment, animals were treated systemically with amoxicillin and clavulanic acid to avoid oropharyngeal co-infection. We evaluated that Paclux was resistant to this combination. Next, the animals were anesthetized, intubated, and connected to mechanical ventilator. After inserting a different catheter for injection and sampling, animals were infected with a sublethal dose of Pseudomonas aeruginosa Paclux under bronchoscopy. The bacterial suspensions contain 1.5 10 bacteria, and it was delivered directly into the right lung. Animals were monitored along the experiment. At the time of the sacrifice, we collected the bile and the lung tissue for analysis. The blood samples were used to assess bacteriemia. The urine sample for and we also analyzed all along the experiment clinical and biochemical parameters. We measure the bacterial load both in the bile and in the lung tissue. The pigs challenge with Paclux following this procedure ultimately developed a fatal severe experimental pneumonia with a clinical symptom, fever, progressive deterioration of gas exchange, and a positive microbial culture. Also, although bacteria were detected in both the right and the left lung, the pneumonia was focalized, limited to the right lungs, with a marked lung inflammation characterized by a massive polymorphonuclear leukocyte infiltration. And we also observed non-aerated peripheral lung tissue segments at the site of bacterial inoculation. No bacteria was detected in the systemic circulation, neither in the urine, which is consistent with a human VAP, and there were mostly no secondary infection. For pilot experiment, animals received the phage twice at 2 and 11 hours post-infection. Control animals did not receive any treatment. Because the mean of animal died after 22 hours, we sacrificed during the development of the Phage model, we sacrificed all the animal at 21 hours after the beginning of the experiment. At this time, the animal of the two groups survived the infection. We collected the bile and the lung tissue for analysis. As previously, we monitored along the experiment. We monitored the animal along the experiment and collected different biological samples for microbiological and histological analysis. Here are the result of the pilot experiment with phage therapy under inhalation. Phage-treated animal showed a very nice decrease of the bacterial load, both in the bile and also in the lung tissue, suggesting that inhaled phage therapy was able to contain Pseudomonas aeruginosa pneumonia. At the time of sacrifice, Phage were relatively homogeneously distributed in the right and the left lower lungs of the treated animals, and they were plentiful of phages in both the tissue, the lung tissue, and in the airways. No infective phage was detected in the sera and in the urine along the experiment. Overall, in conclusion, we found that the nebulization lead to 25 to 60% phage viability, and which is compatible with therapeutic dose for inhalation as the phage can self-replicate. We found that nebulization was very stressful and leads to phage fragmentation, in particular for myoviridae. Maybe there can be some way of trying to improve phage stability during nebulization, working on formulation. For the drug and device, for the evaluation in the pig model of VAP, we found a huge decrease of the bacterial load both in the lung and the airway with inhaled phage therapy. Inhaled phage treatment seems to contain the infection. And we also demonstrated that the phage were able to replicate in situ in the pneumonia fragments. To finish, I would like to thank all the persons that contributed to this work in my group and outside the lab, in Ferricides Pharma and in DTF that participate extensively to this work. And I would like also to thank our sponsors. Thank you for your attention.